Thank you for watching our first video. I'm the Business Development Manager at eEnzyme, and I'd like to introduce you to our ACTOne technology. The ACTOne technology is a robust cell-based assay for G protein coupled receptors. And for the slideshow, we will call these GPCRs for short. We've also applied the ACTOne technology to phosphodiesterases, but we won't be discussing those today. So what's interesting here? About 1/3 of new drugs target GPCRs. So GPCR assays, like ACTOne, have almost become synonymous with drug development. At our last look, only 75 of 500 non-sensory GPCRs have been targeted, but 3 to 4 new potential GPCR drug targets are identified annually, and new response profiles are sought for old targets. Annual sales exceed $100 billion for these drugs. In this schematic of the ACTOne assay, you see GPCRs, once triggered by their ligand, transduce a signal to adenylate cyclase to generate cyclic AMP. The cyclic AMP triggers our patented cyclic nucleotide-gated ion channel. We call it the CNG. The ions generated from the CNG response target a dye reporter molecule, which creates a fluorescent signal. The CNG activity can be regulated by either cyclic AMP or cyclic GMP. Our patented ACTOne CNG is engineered to make it very sensitive to cyclic AMP concentration changes. The data shown here reveal the affinity increase to cyclic AMP with either 1 or 2 key amino acid mutations as compared to the wild-type CNG. A glutamate to methionine mutation at position 583, and that same mutation combined with a change of cysteine to tryptophan at position 460. The ACTOne assay is simple. An easy, homogeneous protocol makes it high-throughput screen ready, and it uses standard lab equipment for fluorescent protocols, giving you easy access to its features. Given fixed R&D budgets, its generally lower cost will allow you more progress. New assays never require GPCR engineering, since it uses the native receptor. And even more so, many cell lines for critical GPCR assays are already established. Yet the ACTOne assay is powerful. This is a live cell, real-time assay with a high degree of flexibility. Just looking at data output, you can have kinetic data, endpoint assays, high-content screenings, or potentially measure the agonist and antagonist action on the same cells. That means cells in the same culture well. Almost all GPCRs have proven amenable to assay. Finally, it generates highly reproducible data across experiments. Here we focus on the protocol ease, showing protocol schematics for kinetic and endpoint assays. Starting with cultured cells, the kinetic assay breaks down to 3 steps, and the endpoint assay breaks down to a 4 to 5 step protocol. Each has their advantages. The kinetic data is useful for assay development with fewer false positives. The endpoint assay has a low instrument requirement and is useful for high throughput screens. Here we show representative data for 2 drugs, isoprotenolol assessed using the kinetic assay, and NECA assessed using endpoint assays with the ACT1 beta-2 adrenoceptor cell line. The kinetic data reveals the fast response time, while the endpoint assays reveal the stable signal produced using this technology. The fast response and stable signal seen here are generally representative of this technology. Comparing the ELISA assessment of cyclic AMP to ACT1 assays reveals the excellent and similar sensitivity of ACT1 technology in endpoint assays for 2 drug compounds, NECA and forskolin. ELISA assays are considered the gold standard assay for cyclic AMP. However, we should mention they are less flexible and limited to endpoint assays. Comparison of FRET assays and ACT1 assays in these experiments reveals similar results for 5 drugs. Looking at error bars, you can see that ACT1 is more reproducible than the common FRET assay, and yet it is an easier protocol. Also note that the discoidin domain receptor 2 assayed here is from the GI family, indicating the robust spectrum of GPCRs assayable using ACT1 technology. Again, a highly flexible assay. This is in-user data where they've analyzed antagonist and agonist in the same experiment, that is, looking at two GPCR modes in the same well, a neat trick. This is enabled by the fast response time and stable signal from the assay, and we are unaware of any other GPCR assay that can do this, again revealing ACTOne as a highly flexible technology. Here are data from a high-content screen using the ACTOne technology, revealing how it can allow imaging of individual cells for this purpose. It also reveals the rapid development of fluorescent signal in the ACTOne assay. There are simply very few GPCR assays as flexible. Using one cell line for multiple assays can allow for lower cost and more comparable experiments. Concerning experimental comparison, these data reveal the excellent signal-to-noise and reproducibility from ACTOne assays. A set of kinetic assay experiments were performed for the data on the left and a set of endpoint assays for the data on the right. The fluorescence increase is compared in the bar graphs. The results were then put through the statistical analysis to evaluate the reproducibility. The results are shown in the rightward boxes. This assay has been widely used by our many customers for good reasons. Here are the summary takeaways unpacking the many reasons that you might use the ACTOne assay on your GPCR of interest. We've emphasized its unique flexibility to underscore the robustness of this technology. This flexibility allows ACTOne technology for the assay of phosphodiesterase as well, although this video does not delve into that application. Finally, thank you for making it through our video. We tried to keep it to an appropriate length and depth. The best way to learn more is to reach out to us, and we can provide either technical advice or a list of publications. Also, we do provide services as well as selling established cell lines for your in-house use, and we'd appreciate the opportunity to help you out.